Welcome everyone to OsteoBites, and thanks so much for joining us today. My name is Christina Iptoma, and I am mom to Osteo Angel Dillon and Director of Scientific Programs at MIB Agents. And today on OsteoBites, we are joined by Dr. Ali Cihan from Memorial Sloan Kettering Cancer Center to discuss his work on targeting EPHA2 with dual armored CAR T cells for immunotherapy in pediatric osteosarcoma. And MIB Agents is proud to support this work with the 2025 Outsmarting Osteosarcoma Hope Young Investigator Grant because of Isaac. Thank you so much, Dr. Cihan, for joining us on OsteoBites today. And everyone, please feel free to submit questions during the presentation through the Q&A function in Zoom, and we will share those questions with Dr. Cihan after his presentation. A little bit more about our guest today. Dr. Ali Cihan is a research scholar at Memorial Sloan Kettering Cancer Center, where he focuses on immunotherapy with a particular emphasis on advancing cellular therapies for pediatric solid tumors, including osteosarcoma. He received his Ph.D. from the Rockefeller University, where his research explored how chromosomal abnormalities and transcriptional dysregulation contribute to the development of high-risk pediatric leukemias. Motivated by the urgent need to improve outcomes for children with cancer, especially those facing limited treatment options, Dr. Cihan turned his focus to bridging laboratory discoveries and clinical application. His current work aims to develop immune-based therapeutic strategies that address the unique biological and clinical challenges of pediatric cancers. We are thrilled to have you on OsteoBites today, Dr. Cihan. A couple of announcements before we get started. Registration for our Factor 2026 Osteosarcoma Conference, June 25th to 27th in Columbus, Ohio, is now open. I'll put the registration link in the chat where you can find more info. You can also find the scientific program and our fantastic speaker lineup. Dr. Cihan will be there, giving more of an update on this work as well. We have a limited number of travel grants for both scientific attendees and Osteo Warriors. I'm going to put a link in the chat where you can find more info and apply. But you have to check it out soon. The Scientific Travel Award applications are actually due tomorrow, March 27th in Proposal Central. Osteo Warriors must be nominated by a healthcare professional, and those nominations can be submitted on a rolling basis. So a little bit more time for the Osteo Warrior Travel Awards, but if you're interested in applying for a Scientific Travel Award, the due date is tomorrow. So with that, let's get started with our presentation today.
Okay, thank you very much, Christina, for your introduction. And as you mentioned, today I will talk about CAR T cell therapy that we are developing by targeting EPHA2 for pediatric osteosarcoma. And I don't have any financial disclosure. And I want to start by thanking MIB Agent to support me and this project by outsmarting Osteosarcoma Young Investigator Award because of Isaac. Unfortunately, Isaac passed away several years ago, but thanks to his contribution, we hope to develop this CAR T cell therapy that will, in effect, help future osteosarcoma patients in need. And here, this will be an outline of my talk. First, I want to remind very briefly current landscape of the osteosarcoma treatment, and then I will talk about the overview of CAR T cells and CAR T cell therapy and the challenges that CAR T cell therapy faces in solid tumors. And then I will talk about the criteria that makes a target ideal for CAR T cell therapy, and then show our functional analysis of EPHA2 CAR T cells that we have developed, and finish with the future directions. So as we all know, osteosarcoma is the most common primary bone tumor in children, and thanks to the introduction of the systemic chemotherapy in 1960s, we have an increased overall survival of the osteosarcoma patients. However, metastatic and relapsed osteosarcoma patients still have very low survival rate, and even if they survive, it's usually not without any side effect or limb salvation surgeries. And unfortunately, since 1960s, we don't really have a successful drug development for osteosarcoma patients. So if we were diagnosed with osteosarcoma today, we would get exactly the same treatment that people were getting 60 years ago. So clearly, there's an unmet need for novel targeted therapies for osteosarcoma patients. And one general approach that others and our group has taken is to look into cellular therapy strategies, including CAR T cells. And as we all know, cellular therapies are using immune cells, and we can categorize immune cells into two. We have an innate immunity system, immune system that utilizes cells such as macrophages, natural killer cells, dendritic cells that provides non-specific rapid response. And on the other side, we have adaptive immunity that relies on mainly B cells and T cells that provide specific long-term immunity. And many of these immune cells are being used in active area of research for the cellular therapies. For example, we have CAR NK cells, CAR macrophages, and more recently, CAR monocytes. We also have gamma delta T cells, and of course, we also have CAR T cells. And T cells, we can also categorize them into two. T cells mature in thymus, and based on the T cell receptor that they are expressing, it can either develop into mature helper T cells that usually functions in conjunction with other immune cells, or cytotoxic T cells that specifically can target and kill tumor cells. And as you can guess, a CAR T cell approach is mostly relying on these cytotoxic T cells. And what is CAR T cells and how we are making it? Basically, it stands for chimeric antigen receptor, that we will talk about this receptor in a minute. So how we are creating this CAR T cell is we introduce this gene into normal T cells by using usually viral vectors. And this T cell start expressing this gene, and this gene turns into a transmembrane protein where it has both extracellular and intracellular region. In the extracellular region, our CAR molecule is recognizing the antigen that is found on the cancer cells. And in the intracellular region, it has several signaling domains. Naturally, in T cells, these signaling domains are present in actually different receptors. And for full T cell activation, we need more than one of these signaling domains to be activated at the same time. This is basically a safeguard so that T cell doesn't go and kill all the cells that they recognize. However, in the CAR T cells, what happens is, just a second. In the CAR T cells, what happens is, since all these signaling domains are merged into one single polypeptide, this antibody-antigen recognition is enough to fully activate T cell. And in turn, what this T cell does is it starts secreting certain molecules, such as perforin and granzyme, and then these molecules will kill the targeted tumor cells. And how does a CAR T cell therapy works in patient? Basically, when a patient comes to hospital, first we collect the blood by leukapheresis, and we isolate these T cells, enrich them, and then activate them. And then we express these CAR molecules, usually using viral methods, and then expand this CAR expressing T cell, which are also called CAR T cells. And then we isolate these CAR T cells from Petri dishes, and then re-infuse inside the patient. And then we expect this CAR T cell will go and find the tumor cells and kill them. And here, this vein-to-vein process takes around 7 to 10 days, thanks to MSK Biotherapeutics core facility that we are actively collaborating. Currently, we have several projects that are either starting clinical trials or in the middle of the clinical trial. And this is we are utilizing from Biotherapeutics core facility and their GMP compliant manufacturing ability. And when this project finishes the preclinical research phase, hopefully we will start a clinical trial with Biotherapeutics core facility as soon as possible. And what about the timeline of these CAR T cellular therapies? As you can see, although this technology existed since 1992, the first clinical trial didn't start until the introduction of second generation CARs. And we will talk about the generational differences in a minute. And also, since 2017, we have so far 7 FDA-approved CAR T-cell therapy, and they are all for hematological malignancies. So basically, CAR T-cell approach was revolutionary for liquid cancers. However, their efficacy in solid tumor were rather limited. And there are reasons that this is the case. First reason is trafficking and infiltration problem of the CAR T cells. When CAR T cells are in the body and in the bloodstream, it has to go through and infiltrate a dense extracellular matrix and fibroblast-rich stroma that are usually surrounding the solid tumors. And so basically what CAR T cell need to do is it must exit the circulation and then cross the abnormal tumor vasculature of solid tumors and then penetrate this dense stroma to reach tumor cells. So it is hard to do for CAR T cells. And even if it passes and infiltrates, there is a very immunosuppressive environment inside the solid tumors. Basically, there are other immune cells such as Tregs that expresses these cytokines. These are signaling molecules that create a repressive immunosuppressive environment. When T cells encounter with these signaling molecules, it actually causes T cell to be exhausted and it stops proliferating and it is not active anymore. And another reason that the CAR T cell is ineffective in solid tumor is we have tumor heterogeneity and antigen escape possibility. Basically, we are targeting by CAR T cells a tumor-associated antigen. But usually solid tumors have a very heterogeneous population of cells. Some cells will express our antigen in a very high amount. However, some of them might not express it at all. Therefore, when CAR T cells kill the antigen positive cells, the leftover antigen negative cells can repopulate and cause relapse. So this is another issue. And lastly, we have cytokine release when a lot of CAR T cells are injected or they start secreting a lot of cytokine. This can also cause cytokine release syndrome. That's a safety issue for the CAR T cell therapies, although recently this becomes more and more manageable by physicians in patients. And our approach to overcome these challenges are basically using armors. And we will use IL-18 and light armor that I will explain in a minute. But to appreciate the armors more, first I will talk about the different generations of CARs. So we have first generation of CAR which only has one signaling domain, which is CD3 based on CD3 zeta, and this is naturally found in the intracellular region of the T cell receptor. And this is usually by itself not enough to activate the T cell. That is why the first generation CARs were unsuccessful. However, when the costimulatory domain is added to the CAR construct, it basically merged two necessary signals into one polypeptide, and by itself, this CAR, after recognition, can fully activate a CAR T cell. So we have two costimulatory domains in the T cells. One is CD28, the other is 41BB. And the third generation CARs included both of them together. However, this didn't have much effect, much increase for the efficacy. But the real difference came when the fourth generation CAR introduced the armors. Basically, armors are these immune modulatory regions. When it is expressed inside the T cell, this will cause cytokine secretion. And these cytokines are usually create a pro-inflammatory environment so that the immune cells that are around in the surrounding, when they are activated by these cytokines, they will activate itself and start killing tumor cells more efficiently. And we will have two armors, IL-18 and LIGHT. Basically, IL-18 is the interferon gamma inducing factor. When CAR T cells start expressing IL-18, it promotes an immune activation, and all the immune cells around that is activated by IL-18 will secrete more cytokines. So this will promote a very pro-inflammatory environment, and in turn, this T cells and other immune cells will be more active and will clear the tumor cells more efficiently. And one other thing that IL-18 will do is it will autoregulate the CAR T cell, and then this will cause CAR T cell proliferation. So it will persist in the body in longer term and can kill tumor cells more efficiently. And the interferon gamma response is shown to be important and actually essential for CAR T cell killing in solid tumors. And here at MSK, we have two ongoing clinical trials that we are using IL-18 as a part of the construct. And from those clinical trials, we can clearly say that it is safe to use in patients. Some patients show cytokine release syndrome. However, it's quite manageable. So it is overall safe to include in our construct. And the other armor that we will be using is called LIGHT. LIGHT is a tumor necrosis factor family member, and it's a transmembrane protein, and it interacts with a protein called alpha-beta R. And this alpha-beta R is actually highly expressed in a lot of tumors, solid tumors. So basically what we have is we have a CAR that will interact with our target antigen, and we also have a LIGHT molecule that will interact with the alpha-beta R that are found on the tumor cells. So what this will create is, even if our tumor cells are antigen negative, they will express alpha-beta R, therefore will be targeted by CAR T cell. Also, this LIGHT will enhance the CAR T cell cytotoxicity, and therefore it will increase the bystander killing. So it's not only killing the tumor cells, it also kills the cells around the tumor cells. And it will also improve immune cell filtration by normalizing the tumor vasculature inside the solid tumor environment. And it also, when it interacts with the tumor cells through alpha-beta R signaling pathway, it will downregulate the anti-apoptotic genes in tumor cells. So this will basically facilitate tumor cell go through the apoptosis and kill itself. And besides this, it also will recruit more immune cells by interacting with other receptors that are found in antigen-presenting cells, such as NK cells and dendritic cells. So it will overall increase the recruitment of these immune cells on solid tumors, on our target, and enhance the cytotoxicity. And I mentioned that this alpha-beta R expression is high in tumors. So when we look at our osteosarcoma patients, this basically shows the alpha-beta R gene expression level in our clinical cohort. These are more than 500 pediatric cancer patients that were treated at MSK Department of Pediatrics. And here you can see that alpha-beta R gene expression is specifically overexpressed in osteosarcoma patients. So basically, osteosarcoma is a very optimal model to incorporate this light molecule into our CAR T cells. And we have this CAR molecule. Sorry, we have this light molecule that will interact with L to beta R, and we have this CAR molecule that will target our target antigen. So for best activity, we of course need to find the right target. And thinking about how we will approach this and apply this immunotherapy to osteosarcoma, we also wanted to be strategic about it. And among the target list, we decided on focused on EPHA2 based on its enrichment in osteosarcoma cells. So what makes EPHA2 or any other target a right target? So basically we have some criteria for ideal target in solid tumors. Well, we first want them to be high differential expression. So we want our antigen to be expressed on malignant cells, on tumor cell surface. However, it has to exhibit low or very restricted expression in normal healthy tissues. Also, we want them to be on the cell surface so that it is accessible by the CAR T cells. And we also want it to be functionally important for the tumor cells, either for its identity or maintenance of the tumor cells, so that it cannot be downregulated very easily and we can keep killing the tumor cells. And now we will go into more details into this criteria. First, high differential expression of EPHA2. So it's not only us, but others have also shown that this is basically a mass spectrometry-based surface proteomics. It shows that EPHA2 is the top upregulated cell surface protein in osteosarcoma cells compared to osteoblasts. So compared to normal cells, EPHA2 is very highly expressed in the osteosarcoma, in the tumor cells. And based on our patient expression data, RNA expression data, we can again see that in our clinical cohort, EPHA2 is highly expressed in osteosarcoma patients. And we also have these patient-derived xenograft models that are based on these patients. And among these PDX populations in the PDX cohort, we again see the osteosarcoma PDXs have the highest level of EPHA2 expression. And this is at the level of RNA. So what about the protein level of expression? Because at the end, we have to target the protein. And here, this is the immunohistochemistry results of our PDXs. Basically, we are using an antigen to visualize the specific proteins in tissue sections. And here, the brown color represents the EPHA2 expression, and a blue color represents the cell nucleus. Here, you on the left side, you see our normal tissues, liver, kidney, spleen, and lung. And on the right side, we can see our PDX, osteosarcoma PDXs. So we have analyzed more than 55 osteosarcoma PDXs. And out of these more than 55 PDX, we actually observed 88% of them have moderate to high level of EPHA2 expression, as you can see here on the right side, and only 12% had low or negative level, non-expressing of EPHA2. So it is in EPHA2, you can clearly see that highly expressed in majority of the osteosarcoma patients. And here, one other thing to comment is I think you can appreciate, especially here on the very right side, the EPHA2 expression is mostly on the cell surface, which is important, and this will take us to the second criterion. And
Dr. Cihan, sorry, quick question. On those OXPDX, are those, is that consistent across primary bone tumor and lung?
Primary, so you mean the metastatic patients?
Yeah. Are these all metastatic? Are these all lung samples or?
No, they are not. It is actually the location can change.
Mhm.
Some are primary, some are from metastatic region. It's basically all PDXs we have. We didn't select a portion of it to test EPHA2 expression. We checked all of them. Some are pre-treated, some are post chemotherapy, treated. Uh, so with the map, so it is a actually combination.
Okay. So you see it it's consistently overexpressed.
Yes,
Okay.
yes. It's not only in metastatic patient.
Got it. Thank you.
Mhm. And for our second criterion, this is the surface accessibility and abundance. We did check the EPHA2 expression using flow cytometry. Basically, this is a method that will use antibody to test the EPHA2 expression levels in viable and intact cells. And here we use two osteosarcoma cell lines, cells2M143B. And here you can see that EPHA2 expression is quite high in osteosarcoma cells compared to their controls. And also we have included a non-small cell lung cancer cell line here, which is also another cancer that EPHA2 is highly overexpressed. And here leukemia is just used as a control. They don't express EPHA2. And here, one thing that I want to point out is we actually use non-small cell lung cancer here because this project is carried out at the same time in osteosarcoma and lung cancer models. And there are reasons for that. Well, first of all, there are much more lung cancer patients, and every year we only have, well, it's actually both fortunate and unfortunate that we only have 1,000 new osteosarcoma patients. However, we have 200,000 new non-small cell lung cancer patient every year. So this will basically both increase the impact of this CAR T cellular therapy, and also it will facilitate the development of the therapy. And third criterion is a functional oncogenic role. And as I mentioned previously, that we want our target to have an oncogenic role either for important for the maintenance of the tumor cells or the identity, so that this cannot be easily downregulated. And here you are seeing in normal cells, EPHA2 is normally expressed, and there is always ligand-dependent signaling. And when this signaling pathways are active, this cell will not basically proliferate or migrate. However, when EPHA2 start to become overexpressed, it activates ligand independent pathways, and in turn what this signaling pathway does is it increases the cell proliferation and cell migration. And it is reported in the literature that EPHA2 expression level is correlated with the metastatic status of certain cancers. And so then if we target EPHA2, and if the EPHA2 is important for tumor cells, they will not be downregulated very easily. And this will allow us to use combinatorial approaches if CAR T cell therapy doesn't show the efficacy that we expect. And so basically, EPHA2 is a very good target, and we are not the, of course, first group that is targeting EPHA2. Previously, EPHA2 is targeted in both clinical and preclinical research. So here on the left side, you are seeing in a clinical trial for glioblastoma, which is another cancer type that EPHA2 is highly expressed. You are seeing a patient, and after infusion, 4 weeks after infusion of EPHA2 targeting CAR T cells, you can see the decrease in the tumor volume in the brain. And in this study, EPHA2 targeting 4H5 CARs are used. So basically, this 4H5 is an antibody developed against EPHA2, and basically every other group are using 4H5 CAR in their preclinical and clinical research. So that is why we will use 4H5 as a comparison to our CAR T cell constructs as well. And here on the right side, you are seeing a preclinical research that's the same CAR 4H5 is being used as a CAR NK approach against for sarcoma, and it shows activity in sarcoma bearing mice, and you can see that it basically suppressed the tumor growth in the mice that is injected CAR NK cells. So this will take, all this information will take us to our hypothesis. Basically, what we are hypothesizing is targeting EPHA2 with novel antibody selection and also incorporating light and IL-18 armors will basically lead to enhanced therapeutic efficacy against osteosarcoma. And to test this hypothesis, we are focusing on two aims. First, functional analysis of our CAR T cells. These are basically in vitro experiments that we will analyze. And then we will evaluate those CAR T cells in vivo with basically using mice studies. And in first specific aim, we will analyze these CAR T cells functionally. Basically, what we are doing is, well, here I'm not going in a lot of detail, but we generated novel antibodies against EPHA2 by using a special type of mice, and these are all humanized antibodies. So we selected the top 12 of them and converted them into a CAR construct. So we engineer our CAR using these 12 antibodies. And we are testing these antibodies in either cytotoxicity assays or proliferation assay. Basically, cytotoxicity assays are using our effector cells and test how effective the tumor cell killing, target cell, in this case, it's tumor cell killing in vitro. And we also test using a proliferation assay where we use real-time live cell imaging to continuously monitor the proliferation, cell growth, and CAR expression of our CAR T cell construct over time. And based on this cytotoxicity and proliferation assays, we have identified our leads. So this is very simplification of many proliferation and cytotoxicity assay in all these experiments, or rather than showing 100 different figures and the graphs. So in each experiment, basically I measured the efficacy relative to 4H5, considering 4H5 as 1, and then ranked our binders based on their efficacy. And here you can see, and we also use non-targeting CAR as a negative control, of course. And here you are seeing the, well, for the sake of simplicity, I numbered them 1, 2, 3 based on their efficacy. So these 3 CAR T constructs will be used against 4H5 to compare in our in vivo experiments, basically. And before going into the more detail for the in vivos, I want to show one in vitro result so that you will have some idea how well they are killing the tumor cells. So here you are seeing the cell killing assay by increasing amount of CAR T cells. And here we have non-targeting CAR T cells in blue, and we have 4H5 in red. And here you are seeing the percentage killing is higher for our CAR T cells compared to 4H5. So it is our CAR constructs are performing better than 4H5 in osteosarcoma models and also in lung cancer models. And here you are seeing 1 and 2 is better than both 3 and 4H5 and also M-Cherry. And then I tested, of course, these CAR constructs in vivo studies. And basically we use two separate models. One is CDX models, where we are using cell lines and grow them in this immunocompromised mice that allows a human cell growth in it. And we also have PDXs and grow PDX in mice as well. And here I will show our data for our CDX models, where basically we injected tumor cells, and after 1 week we injected our CARs for 5 different group of CARs, and we imaged the mice every week to check their tumor burden. Here, since our tumor cells are marked with the luciferase, we can actually visualize them. And here the color represents the amount of tumor that they have. And here you are seeing in our non-targeting control, week by week, our tumor burden is increasing in all mice. And here for H5, what are basically competitor, what everyone else is using against EPHA2. And you are seeing that it actually has some activity and it is decreasing the tumor burden in mice. However, if you look at our CAR-T number 1, you will clearly see that this is performing much better to suppress the tumor growth against 4H5 or also in our non-targeting control. Here, of course, number 2 and number 3 are also performing well, but it's clear that number 1 has the most activity. And this was in one of the model. This was actually lung cancer model. And these CAR T cells are not only suppressing the tumor growth. If you look at the survival data, here, this is a Kaplan-Meier graph, and this are basically the percentage of survival is decreasing every time a mice dies in that cohort. And we usually compare the median survival, the 50% of when 50% of the mice dies. So here you can see in these models, actually both CAR-1 and CAR number 2, when we stopped the study at day 200, they actually more than 50% of the mice were still alive in both groups. However, median survival day is for non-targeting CAR is 105 days, and for 4H5, 114 days. So it's clearly that our CARs are prolonging survival in this model. And of course, we observed similar things in osteosarcoma models as well. And here you are seeing this is our non-targeting control, and here our 4H5. So here, I want to mention that this actually 143B is much more aggressive model than A549, our lung cancer model. And usually it starts to kill mice around week 5. And you can clearly see that actually more mice is that in much faster in this study. But again, we can confirm the efficacy of our number 1 CAR against 4H5 and non-targeting control. And here, median survival days is, if you look at this orange and compare it with the blue and the red, you will see that compared to both non-targeting CAR and the 4H5, they are increasing the prolonging the survival significantly. And also another thing that I want to mention is actually these are all second generation CARs that we haven't even incorporated our armor yet. So when we incorporate our armors, IL-18 and light, this can only go in a better direction, and it will be our CARs will be more efficient at killing tumor cells. And right now, basically, we are doing this, optimizing our lead CAR-T number 1 construct. So this CAR construct has different modules, and we can optimize and modify each of them to enhance the activity of CARs. So what basically we are doing is optimizing the linker sites between heavy chain and light chain, the antibody region where it recognizes the EPHA2. And we have 7 different versions of these CARs right now by using the same antibody, but just increasing the linker size. And we also have IL-18, basically armor incorporated version of it. And we are testing all of them in the in vitro and in vivo. And here I will show the in vitro experiment, both using 143B osteosarcoma cell line and also in our lung cancer model. And you can see that actually all our CAR-T constructs are effectively killing both osteosarcoma and lung cancer model. And currently our in vivo experiments with both osteosarcoma model and lung cancer model is ongoing. And hopefully I will update everyone about this in Factor meeting. And the future directions basically will include first to finish the functional analysis of this CAR-T cells in vitro. Basically, the only after optimizing this construct and finding the lead candidate, our top candidate, we will only include both armored CARs and basically finish our in vitro experiments. And also we will evaluate their in vivo activity by using these CARs, using different models. We will continue using this aggressive osteosarcoma models. And also we will currently expanding our PDX models that have different varying degree of EPHA2 expression. And when we have enough PDX cells, we will start these experiments as well. And these are basically to test the efficacy of our CARs. And based on efficacy, of course, we want to check the safety of our CARs as well. And for safety, we are using humanized mouse models. Basically, in this model, we have an humanized EPHA2 protein in mice so that we can test our CARs if it is creating any toxicity for the normal tissues. And lastly, we have syngeneic mouse models that we are actually soon start testing our mouse osteosarcoma cell lines and mouse CAR-T cells, and we will inject these both cell line and CARs into our immunocompetent the mice that has normal immune cells. It's called the Black 6 mice to finish the safety evaluations as well. And to summarize the key points, basically osteosarcoma urgently needs new targeted therapies, and we believe EPHA2 is a very strong candidate. It highly expressed in osteosarcoma, and it has a very limited expression in healthy normal tissues. And we were able to confirm EPHA2 over expression at the RNA level and at the protein level in patient samples and PDXs. And our EPHA2 targeting CARs demonstrate better activity in vivo. They suppress tumor growth and also prolong survival in both osteosarcoma and lung cancer model. And we believe that EPHA2 is compelling target that supports the development of CAR-T cellular therapy. And of course, I want to acknowledge everyone that's involved in this project, especially all members of Kang de la Cruz Lab, led by Andrew Kang and Phil de la Cruz, and Daniel Lab members, led by Tony Danian, and Pediatric Cell Therapy Program, led by Kevin Kern. And thank you for your attention, and I'm happy to take any question.
Thank you so much, Dr. Cihan, for that great overview. And actually, before you make your slides go away, we had a question that came in, I think that was related to your slide a few back where you were showing the 143B OS line, maybe that one, I think. Yeah. So there was a question that came in just asking how you injected the tumor cells and after how many days from tumor implantation did you start therapy?
Mm-hmm. So these are actually in vitro. So I think maybe it is related to this in vivo study. Basically, we injected 100,000 143B cell line at day 0, and CAR injection happened 3 days after. And this is since it is very aggressive model, we cannot wait 1 week for this model, because if we wait, we can definitely not control tumor cells. If they already overgrown the CAR T cells in the at the, like, if you look at the sheer numbers, it's more than probably like 10,000 times more. So that's why they cannot catch up and kill tumor cells efficiently. And in this one, we injected CARs 3 days after tumor cell injection. And every week we basically imaged, but I'm only showing, of course, certain weeks for the sake of simplicity.
And follow-up question to that, for that injection, was it IV or peritoneal?
IV. So this, we created the metastatic model. Yeah, maybe I forgot to mention that both models, since we really want to start clinical trial in metastatic patients. So it is more relevant to us, metastatic models. So all the models that we are using and we will be using very likely be metastatic models. We wanna show the efficacy in metastatic models.
Great. Everyone, feel free to add any questions that you have to the Q&A, and we will make sure to ask Dr. Cihan. But I have a question that will take us back to one of your first slides on the role of innate versus adaptive immunity in immunotherapy. And you mentioned that the CAR T approach was really leveraging the adaptive immune system. But I was wondering if you could talk a little bit more about how the two relate and kind of the role that they both play in cancer.
Other cells?
Yeah. Like,
yes.
There's one that's more important.
Well, all of them are important, obviously. But basically, the main differences is this adaptive immunity is MHC restricted. So actually, this is only can be applied as a autologous model. So basically, we have to get the T cells from the patient and modify it and give to the same patient. Of course, there are ways to overcome this, and a lot of people are working on it, but I don't think it is successful. I'm talking about the CAR T cells, specifically. But if we look at the other approaches, they are actually allogeneic models. So you can produce other immune cells and modify them with CAR and can use it for other people, the treatment of the other people as well. So this with what's called the off-the-shelf approach is actually quite popular, and people try to achieve this, because it will be very simple to treatment, and it will also cut costs a lot, because these are not cheap treatments. So overall, is there any FDA approved treatments? Unfortunately, no. So you can see both have advantages and disadvantages. So far, we only know the CAR T cell will work, but hopefully, other immune cell approaches will also work. And our group here, cellular therapy group, are working on try to make other immune cell work as well. So if it is successful, then we will be able to use this off-the-shelf approach, and it will be quite cheap. So it is, of course, it will require some optimization, but when we find our lead construct against EPHA2, we can definitely move this construct into other immune cells, start expressing there to target EPHA2 as well. So this is a strategy that we are considering, but of course, our first and easiest approach will be CAR T. After we show that actually CAR T cell therapy works, that is something that we would like to explore later on.
And for the CAR T, is there an optimal time for that T cell collection or leukapheresis? Because most patients, when they're enrolling right now,
yes,
CAR T clinical trial, they're heavily pretreated, and so
yes,
I'm pretty
I believe as early as possible. before any treatment. Right now, I showed our vein-to-vein timeline in our Biotherapeutics core facility. I said it is 7 to 10 days, but actually it can change from patient to patient and cancer to cancer, because some I know for some clinical trials, I think especially for AML that they are using, if a patient have very low quality cells, if it is being collected after many different therapies, it might not be as good as it like for its potential, so that they have to utilize, unfortunately, low level of cells and low level of CAR T cell. So I'm not sure at the clinical side, at the treatment, how this works, since, I'm not seeing patient, but I would definitely watch for, as early as possible.
Yeah, I was going to say, I know there are other practical concerns like cost and all that stuff, but it seems like, can you manufacture the T cells off frozen samples? Like,
yes.
would you collect like
you can.
prior to the
That is
store it away in a freezer for later?
Yes, that is what they are doing for clinical trials for some basically patient, just because that their I think overall like T cell quality is not as good after many treatment, passing many treatments, and so they go and look for earlier collection and basically use those blood to make CAR T cells for those patients. So it is definitely a possibility.
Gotcha. So that's like timing on the actual T cell collections that you can.
Yeah, the best that every week we get fresh blood, collected blood from New York Blood Center, and it is clear that when it is fresh, when it is processed immediately, we get the best quality and the highest transduction CAR T cells.
Yeah.
But if we wait a little bit long, if we wait several days and then process it, our CAR T cell yield is actually, or T cell and CAR T cell yield is actually decreasing. It's very clear, like in our hand as well. So
yes, it's a delicate balance between having good cells from the patient, but also having good yield.
Yes.
And so what about, you alluded to this earlier about so that's for the collection to manufacture the CAR T cells, but what about optimal time for actual the CAR T cell therapy?
So I think whenever the patients are enrolled in clinical trials, they can start as soon as possible, just because the tumor is already grown and it's growing inside. I think what they do is, for majority of the clinical trials, they give lymphodepletion, just because that lymphodepletion actually increases the proliferation of CAR T cells whenever it's giving, since it basically kills majority of the lymphocytes inside the body. So then body try to recover. So I think it needs to given 3 days before. So technically, even the off-the-shelf approaches, when it is available, I believe after 3 days after lymphodepletion, CAR T cells can be injected in patient. And earlier is the better, of course.
Yes, yeah. Okay, another question came in. It's a good transition about light. So light is a trimer. Have you found evidence you're getting trimeric light secreted from your multistronic CAR constructs?
We don't. We actually have homogeneous light. We don't have LT beta. Those are usually heterogeneous. But for the light, we only, as far as I know, we only have the homogeneous version.
So you did a really nice job.
And it is known that light, we are targeting with light, but it is known that LT beta R is a good target. It is shown recently by other groups as well. It's just that other groups are targeting LT beta receptor using LT beta or LT alpha, and those are heterogeneous ones.
So you did a really nice job kind of laying out the different challenges for using these cellular therapies in solid tumors. And so, if I'm understanding correctly, you're basically trying to address the heterogeneity issue and antigen escape with the L beta R. So
Correct. The light, yeah.
Yeah, if you can talk a little bit more on that. Is it on all tumor cells? Is it on normal cells? I think I saw a chart in there where it looked like it was like 20 to 40% in osteo. And then also you were also I'm curious also about L beta R and other cancers.
Yes. So actually, it is highly expressed in a lot of different type of cancers. In the lung cancer models that we have, or glioblastoma, medulloblastoma, it is expressed as well. So basically, light will normalize the tumor vasculature, and this will facilitate the CAR T cell infiltration, and then IL-18 will increase the proliferation. So we hope to overcome this trafficking and infiltration problem by using light. And as I mentioned, light alpha beta R interaction will also enhance the cytotoxicity, and this will allow bystander killing. Basically, when CAR T cell start to secrete molecules, it will not only target the cell that it is interacting, it will target the cells around it as well. So then we hope believe that at the end, this will allow us to get rid of as much as possible the tumor mass.
And so another question is about the target EPH2. And did you see, you had all the illustrations with the mice, efficacy correlate to your expression at all?
So those are experiments that we have to do still. So right now we are starting our PDX models, and we selected different level of EPHA2 expressing PDXs to test if the efficacy actually will be depending on its expression level. Of course, I'm talking about for the in vivo. We have to do those experiments, but from the in vitro data we have, basically the cell line data, I can tell that even if relatively it is not as high as SAUS2 or 143B, the osteosarcoma cell line that I showed that they have high level of EPHA2, other tumor types that has slightly lower EPHA2 levels, actually our CARs can kill them very efficiently as well. So we believe actually this will be replicated in our in vivo experiments as well. So hopefully, even if EPHA2 levels are not highest, we will be able to kill those tumors efficiently as well.
And so likely as you translate this to clinical trial, it would likely then be open to patients regardless of their expression level?
Well, yeah, so there are a few considerations there as well. So basically, so these are the EPHA2 expression levels in PDXs. So I mentioned, but didn't show this. So for our PDX distribution, as I mentioned, only 12% of the patient have actually either low or negative level of EPHA2 expression. And 88% has moderate to high level of expression. So first of all, to better formulate this in our protocol, I have to test PDXs at the different level of EPHA2 expressing ones and see if our CAR efficacy is changing. And the way I explained before for the other question, currently we don't have in vivo data, but from our in vitro experiments, we expect that even if EPHA2 expression is moderate, we expect to actually clear these tumors as well. If it turns out to be the case for the in vivo, then I think it will be more logical to include everyone in the clinical trial as well, just because that 88 of the osteosarcoma patients already have moderate to high level of EPHA2 expression. So this will be basically determined by the in vivo experiments that we will be doing, but it makes sense to include all also the other perspective that we mentioned that it's both fortunate and unfortunate that we have very limited number of patients usually. So it might not be a good idea to start excluding patients if they can have some effect.
Yeah. And would you be able to quantify, I guess, how much of it is from?
Well, we can from RNA with the RNA-seq, and also we can also use immunohistochemistry method by taking some sample from their tissues. We can quantify EPHA2 expressions, but I'm not sure if we will do it yet. So just because that we haven't started writing the our clinical trial protocol, but it is possible that those will be added as criteria.
Yeah, I actually wanted to ask about quantifying how much of the killing was coming from targeting the EPHA2 versus from light.
It might be. It is. I think very hard. So you mean we can also test definitely for their LT beta R expression, but I think defining which ones are killed by CAR interaction or which ones are killed by light interaction, I don't think it's possible to really tell.
Yeah.
But overall, we want them to, have a synergistic effect so that they both can kill all the tumor cells so it doesn't relapse.
Right. Teamwork makes the dream work.
Yes, exactly.
One last question. Any idea about the EPH2 downregulation in TME after the therapy?
We don't have any information about that yet. I think they're talking about our possible CAR T cell therapy, right? Not the chemotherapy.
Right.
Okay. So it is, that is why we are using light and IL-18. Actually, we would like to target antigen negative tumor cells as well, either through LT beta R or through just being adjacent to the tumor cells that are being killed. So we believe and hope that we will clear all tumor cells, including antigen negative ones, and before EPHA2 is downregulated. And actually, I have to also mention maybe the amount of CARs that we are using in these mouse models are actually less than 1 million. So it is actually quite good number, and we can clear the tumor in some of the models. This is very, very clinically relevant amount of CAR because based on our other clinical trials and other projects that are ongoing, if we can decrease the CAR amount less than 200,000, we think that we will see very good efficacy in patients very fast before they even turn down their EPHA2 expressions.
Yes, and I know we're over, but sorry, that just led me to, I wanted to ask this last question about, so again, in clinical translation, how many infusions do you think you would?
Oh, that is something that, we have to address later by some experiments. So we actually, after safety evaluations, we will do some dosing experiments as well to see that basically the, repeating the CAR T injection will have any effect or will have clear more tumor or more efficiently in shorter time. This is basic, it has to be based on our experimental data, and we haven't done those yet, but those will be done after safety testing.
Okay, so we are at the top of the hour, so I'm gonna not be greedy with your time, and thank you for joining us on OsteoBites today, Dr. Cihan, and making it better for osteosarcoma patients. More information on this and all OsteoBites can be found on our website at mibagents.org, on our MIB Agents YouTube channel, or at your favorite podcast place. And we hope you can join us next week on April 2nd. We are going to be joined by Dr. Ram Rao from Seattle Children's Research Institute, and he will discuss his Outsmarting Osteosarcoma funded research on extracellular matrix matrix degradation to overcome osteosarcoma chemo resistance. You can find our OsteoBites lineup for the next few months on our website. Subscribe to our YouTube channel or follow us on Spotify so that you never miss an episode. And if you have ideas for future topics that you'd like to hear about, please email us at events@mibagents.org. Thank you again, Dr. Cihan, for spending time with us today. And thanks to all of you for taking time out of your day to join us. We hope to see you back here on OsteoBites next week on April 2nd when we chat with Dr. Ram Rao. Thank you. Thanks so much, Dr. Cihan.
Thank you very much for having me, and I hope to see everyone in Factor meeting June.
Great. Thank you. Bye-bye.